Thank you. Good morning. My name is Karla Paniagua. I'm a PhD candidate at UTSA, and I will be presenting you today my research. It's called Dissection of the Tumor Microenvironment using Spatial Transcriptomics and Deep Learning. So, well, just a brief introduction. As we already know, cancer cells are not going to be alone in the tissue. They are going to be surrounded by other cells, including immune cells, fibroblasts, endothelial cells, and even extracellular matrix molecules that are going to be influencing and contribution to the tumor progression and modifying the tumor microenvironment. This can have a tumorigenesis effect and also a treatment resistance. So there have been previous studies that have already been working with the tumor microenvironment in cancer, which can be very promising for generating new therapeutic targets and developing personalized treatment strategies. But still, understanding this tumor microenvironment composition is a challenge, and well, there's still a lot of work to do. So, in our research or in our project, what we did is to generate a pipeline that we call transformer and a graph autoencoder to identify tumor microenvironments, TGME. And basically, this pipeline has a set of processes and deep learning model that includes a transformer model and a graph variational autoencoder to identify or to dissect the tumor microenvironments in spatial transcriptomics data, which, well, spatial transcriptomics data, for those of you that have not worked with it, it's going to give us the expression of the genes, just a single cell, but it's going to also provide the spatial location of the cells and the transcripts in the tissue. So talking a little bit about the pipeline, let me turn on my pointer. Well, this type of data, as I was telling you before, it's going to include the spatial location of the cells, it's going to give us the gene expression of the cells, and it's also going to give us what we know as the morphology images. So our process starts with processing the single cell gene expression matrix, just as we do usually in single cell. We're going to do quality control to remove dead cells and possible doublets, and we're going to go through the process of normalization, principal component analysis, dimensionality reduction, everything, till we obtain the clusters. These data that we work with already have the cell types, but there's also a process that you have to do. You will have to find the cell typing. And then we're going to work with the morphology images. What we're going to do is, since we already know, thanks to spatial transcriptomics and the segmentation data, since we already know the center of the cell and the size of the cell, we're going to go and crop individual images for each of the cell, something like this. So we can also include them in our pipeline and enrich our analysis to get a better dissection of the tumor microenvironment. We're also going to build an adjacency matrix, which is basically going to tell us if two cells are neighbors or not. We're going to be building it from a ball tree graph. We're going to be, as I told you before, we're going to be working with a normalized gene expression matrix. And we're also, since we already know the cell types, we're going to be one hot encoding the cell types to include this information too in our model. Then, with these morphology images that we extracted for each of the cells, we have to pass them through a convolutional neural network to extract their features, because we cannot use the image directly, right? We need to convert them to numbers. So we're going to go and use ResNet50 to extract features of these images, and these are going to tell us a little bit more about the morphology of the images, like size of the image, size of the cell, type of the cell, even, because these images are stained with different markers. And well, they're going to give us a little bit more information of the cells. So after we obtain all this information, we're going to go through a process that we are going to be calling TGME normalization. So TGME normalization, what it's going to do, it's going to modify the gene expression of the cell by accounting the morphology images, the gene expression of the cell, and the neighbor cells, like the adjacency of the cells, and also the cell types. So it's going to take one cell and a neighbor cell, and it's going to calculate a similarity weight through this process. And then we're going to use that similarity weight to modify the gene expression of our original cell, accounting for all this information. So after we normalize our gene expression, what we're going to do is to pass them through a artificial intelligence or deep learning model. Our model is going to be composed of a transformer model and a graph variational autoencoder. I'll give you more details in a bit. We're going to take our matrix, pass it through our model, and we're going to extract embeddings, which are going to be giving us a little bit more information. I'm going to explain this in a bit. We're going to take these embeddings, and then we're going to cluster them to identify the different microenvironments. So after that, we're going to get a set of clusters. We need still to interpret those clusters to see if we find, for example, the tumor, the border of the tumor, some vessels, etc. So for doing that, what we're going to do is look at the cell type composition of each of the clusters to assign, a name to that microenvironment. And then finally, we can do the downstream analysis depending on what you're looking for. For example, we can do differential gene expression analysis, and then we can do enrichment analysis depending on what you want to do. So let's talk a little bit more about our model. So as I was telling you before, our model is going to be composed of a transformer model and a graph variational autoencoder. Our transformer model is going to be trained to identify cell types, but we're not going to care about those cell types. What we're going to care about is the attention scores generated by the embeddings, which are going to be telling us the interactions between the genes in a cell. So we're going to extract that matrix, and then that matrix is going to be part of our input to the graph variational autoencoder with our graph that we generated before in the previous steps. Our graph variational autoencoder is going to be trained to reconstruct like these two matrices. But again, what we're going to care about, it's going to be the embeddings generated by the decoder. These two embeddings are going to be concatenated, and that's what we're going to be passing through our model. So as I was telling you before, the transformer model, it's going to be trying to identify the different cell types. So we're going to have as an input a cell individually, and for each of the genes, it's going to be calculating an attention score, which is what we're going to be what we're going to be working with. So our transformer model, it's going to have 3 attention layers. Each of them, it's going to have 3 attention heads. And then for graph variational autoencoder, we're going to have the input as input the output of the transformer model. Well, when we extract these attention scores with our graph, and it's going to be made of an encoder with 3 graph convolutional layers. And then we're going to have a decoder to train our model. So let's talk a little bit about results. To verify our pipeline and our results, what we use is we work with 3 different benchmark data sets. This one is for brain of a mouse prefrontal cortex, and we have the annotation generated by pathologists. And then we, of course, evaluated our method, and we compared to 3 different methods and lead-in clustering. In all the cases, we observed that our method was the one that got the highest scores. We can see that also we got more clearly the layers and some other methods. Then we also work with a breast cancer. Well, actually, we did 2 breast cancer data sets, but I'm just going to show you the results of one of them. And again, we had the annotations by the pathologist, and we evaluated our method to see how good we could identify the different microenvironments. And again, our method outperformed the rest of the methods. It's the blue one. After that, we also did some ablation studies to see how important it is to include all the data that we're using in our model. So we tried, well, this is the annotation of the pathologist, which we compared with, and our model with everything, all the different data, morphology images, including everything. Then we tried without the gene expression, and we see a big drop of the ARI and NMI scores. We again did the same thing, but now removing the morphology images, the spatial location of the cells, and the cell type composition. And in all of the cases, we identify that our score drops. So it's very important to include all of these different data into our model to have a better prediction of the tumor microenvironment. And then we again did an ablation study, but now to see what happens if we remove one of our models or both of them, again, to see how important it is. And we observe, again, when we only use a transformer, we see a drop in our scores. Again, same thing when we use the graph variational autoencoder only, like without using the transformer, we still see a drop. And then when we don't use any of the models and instead we replace it with a fully connected layer, we can see a drop in the accuracy of our model, but in the ARI and NMI scores, which help us to get a better idea of how good these 2 models are, or how important these 2 models are to identify the different tumor microenvironments. So after we tested our model and we saw that it works pretty well, what we did was to use another data set. This data set comes from NanoString from the Cosmex technology, and it's a non-small cell lung cancer data set. That data set, it's high resolution spatial transcriptomics. We use 4 different samples from 4 different patients with different levels of severity in the tumor. So for the first step, we applied all our process, quality control, dimensionality reduction, etc. And we also integrated the 4 samples together to remove batch effects. Here is the in-view map. We also make sure the cell typing was correct because the cell type was provided by NanoString. So we just did like a fast checkup. And then we just evaluated our clustering results, like how good our data was integrated and clustered. After that, we work with individual samples to extract or to dissect the tumor microenvironments. So the first thing was to apply our pipeline. So after doing the TGME normalization, we observed a very nice performance, an increase of the cluster separation. So the integration of all the morphology images, adjacency information, cell typing, etc., will help us to increase the cluster separation. And you can see in here, these 4 images are our 4 samples on severity order before applying TGME normalization, and then after applying TGME normalization, where we see a better separation of the clusters. And also our scores improved before and after clustering. Sorry, before and after normalization, not clustering. Then we went in and trained our model, and we, in here we're just showing the results of when we train the transformer model. We see that.
Your
Sorry.
time
Okay.
is up. Please to the conclusions.
Yeah. Thank you. So, in this part, we just observed a very good score for the training of the model, and then we see how the accuracy decreases a little bit with severity of the samples. Finally, we got to extract the different microenvironments of the tumor, where we were able to observe the tumor cells in light orange, the border in a darker orange. We observed vessels and some other regions of the tumor microenvironment. And to study the differences between this tumor that we found, because we found different types of tumors in one sample, we applied the differential expression analysis and then enrichment analysis, and we identified that the tumor regions were enriched with different pathways: hypoxia, epithelial mesenchymal transition, apoptosis, and we saw that some of them were repeated across the tumor samples. So just concluding, the TGME pipeline achieved significantly higher ARI and NMI scores compared to other previous methods. And then one of the main advantages of our model is that it can profile individual cells, if it's the case that you have high resolution data. And then the combination of the gene expression data, it's going to provide insights into the molecular activities of the cells, mainly into the genes that are actively transcribed and potentially contributing to tumor progression. The inclusion of the morphology features offers now the structural perspective, including the shape of the cell, size of the cell, and even like type of the cell. The cell type information helps to understand the distinct contributions of the different populations of the tumor microenvironment, and it's going to allow to identify different cell types, specific gene expression changes in the potential implications of the tumor microenvironment. And that's it. Thank you. Sorry.